Now, let me turn to the second point that I wanted to address, which is now that we've established that there's a lot of activity in metastatic cancer, what can we really learn about localized cancers? And can we start to devise approaches to look at early vascular invasion? These data are much more preliminary, so I'll present them much more briefly. And for that, we actually turned to prostate cancer. And the reason for turning to prostate cancer was two. The first is that there's some very unique markers in prostate cancer, PSA, or prostate-specific antigen, being one of them. Women don't have PSA. If your cell in the blood has PSA, it clearly comes from the prostate. And if your prostate was resected surgically, there's no other place that the PSA can come from. So really, it's a very useful tumor to study from the point of view of markers. Secondly, prostate cancer has multiple foci of initial tumor when it's resected. It typically recurs years later with metastatic disease to the bone, which is very difficult to access. So again, being able to access CTCs becomes very important to study these. What we're able to do is to show that women have very few PSA-positive CTCs in the blood, if any. They're shown here in purple. Normal, healthy males have a low number. This is a log scale on the left. And if we look at all patients with prostate cancer, early, late, responsive, unresponsive, we again see this distribution of CTCs in the blood. If we take individual patients with metastatic disease, we see serum PSA or CTCs. Once they're treated with hormonal therapies, we see a decline in their treatment. And if we look at molecular markers, this is the TMPRSS2-ERG chromosome translocation, an RNA level type of diagnostic. We find that we can identify that in CTCs from patients with prostate cancer. And the correlation between the initial prostate tumor and the CTCs in the blood 3 years later, the time of metastatic disease, is approximately 70%. So again, pointing to the fact that you can't just assume that whatever molecular marker was identified at the time of initial diagnosis is going to be the dominant marker years later when the tumor becomes metastatic. So turning to early disease, we set up a collaboration. This was led by Rick Lee in medical oncology, a former postdoc of Bob Weinberg's, with Doug Dow, one of our urologists. And we asked a question of a first set of 19 patients who had localized prostate cancer and all saw the same urologist. What did we see? And it turns out that of 19 patients, 11 of them had no circulating tumor cells at all before or after surgery. You can see CTCs in green. In red, you see the serum PSA level, which was present about 4 before surgery and then dropped, thankfully, after surgery. There was a second subset of patients in whom we actually could find CTCs. So about 8 patients had CTCs the day before surgery. Within 24 hours of surgery, the number of CTCs was down to 0 and it stayed down. So that tells us that there's a subset of patients who actually have cells in the blood with localized disease, no evidence of metastatic disease, and as soon as you take out the primary tumor, these cells are gone with a half-life that's clearly smaller or shorter than 24 hours. And then another set of patients who turned out to have a delayed decline. So everybody's CTCs declined by 3 months, but some of them actually had persistent CTCs even after the primary tumor had been resected. And that seemed to be quite curious. It turns out there's a lot of data in the field about micrometastatic disease. Often in the bone marrow, you can find keratin or PSA-positive cells. So we hypothesized that in some of these patients, some cells actually set up some distant site, whether it's in the bone marrow or elsewhere, which persisted and shed CTCs for a limited period of time after the tumor was resected, and eventually this burnt out. What this means in terms of risk of recurrence is something that we're following very acutely now. So in summarizing this piece, I would say that there's some biological insights that we're starting to get into prostate cancer and early prostate cancer. We have some indication from these resection studies that the half-life of CTCs in the circulation may be quite short, and in some cases maybe less than 24 hours. It brings up, I think, a very important point in terms of the timing of vascular invasion and CTC shedding. The model had been that CTCs are really rare and happen late in metastatic disease because you can't see them very well. In fact, it may be that as the technology gets better, you can see these. They may be much higher in number, and it may very well be that invasive tumors send out CTCs into the blood for prolonged periods of time, even years, before one of them actually sets up shop and becomes metastatic. And if that's true, that's obviously very optimistic for approaches of early detection. And finally, the clinical significance of this, of course, needs to be evaluated. So we're now launching a 200-patient, 2-year study at MGH to look at follow-up in patients with and without CTCs. Now, I want to end with some biological insights that relate to metastases. And again, here, just a few slides because this is very, very preliminary. The two observations that we think that we're particularly excited about. One of them is the rare observation, if you could turn down the light just for this one slide, the rare observation that in some cases CTCs come in clusters. We had not seen this with the micropulse technology. We see this with this herringbone technology, and you can stain them as well with H&E. You can see in red, these are white cells that are part of this cluster, and here you see 12 cells in one big clump that you can see in this patient with metastatic prostate cancer. We see this in lung cancer as well. Again, you can see clusters of these cells. They're not common, but if they're true and if they're really representative, they suggest it's hard enough to imagine that CTCs can get through capillaries in the lung and elsewhere. It's even more amazing to think that there could be tumor microemboli that can be basically circulating for periods of time in the circulation. And the question is, do these have a role in initiating metastases? So again, the question of how does this correlate with other known mechanisms or postulated mechanisms of initiating metastases. The second observation is the fact that we can now start to interrogate individual CTCs at a higher level of resolution. We're increasing the numbers of colors and the numbers of molecular analyses on CTCs. And what you're looking at here are 2 prostate CTCs stained for PSA, but also stained for Ki-67, which is a marker of cellular proliferation. This guy is in proliferation. This guy is quiescent. So what we can now derive is a proliferative index of CTCs. And what was quite remarkable in our early study of prostate cancer is that the proliferative index of CTCs varies from 1% to 90% in different patients. And overall, when we look at patients who are responsive to hormone, it's called castration sensitive, but these are patients who were just diagnosed, respond to Lupron or hormonal therapy, on average, the proliferative index is about 1%. When we look at patients with very advanced disease who no longer respond to hormones and are on chemotherapy, some of them have lower numbers of CTCs in the blood, but the proliferation index, the number of Ki-67 cells, is considerably higher, on average 48%, often as high as 73%. So that really suggests that it's not all CTCs, it's a heterogeneous population, and what we really need to know are which are the proliferating cells within CTCs. So to conclude, I've tried to convince you that there are some applications of CTC biology for real-time genotyping in metastatic disease, detection of early invasive disease, and eventually learning what metastasis is all about in human cancers and hopefully being able to target these therapeutically. This work was a very extensive collaboration. We've essentially merged our labs and our funding and our lab meetings between our own group at the Cancer Center, which is co-led by Shyamala Maheswaran, and the group of the BioMEMS facility at MGH led by Mehmet Toner, with the 2 postdocs who've developed phase 1 and phase 2, Sunita Nagrath and Shannon Stott, and a number of molecular biologists and clinicians all involved with these studies that I'm describing here. And then our funding sources, NIH, Stand Up to Cancer, Prostate Cancer Foundations, and Howard Hughes as well. Thank you very much.